Sanger joined biochemist Albert Chibnall's team of scientists who were studying the amino groups of insulin. This protein hormone was discovered in the 1920s and was of immense interest since it became known that it could be used to treat diabetes. He realized that insulin was made out of two different types of chains, a phenylalanine and a glycine chain. Sanger was able to sequence the amino acids of each chain. He was also able to deduce how the two chains were linked together by two sulfur atoms as SS covalent bonds. And by 1954, Sanger had derived the complete structure of bovine insulin. Then, in 1958, he received a Nobel Prize for his research. Sanger's interest then switched to nucleic acids as he began RNA sequencing. Sanger used radioactive methods for sequencing RNA, and in 1967, the first RNA was fully sequenced, with tRNA being successfully sequenced in 1969. By 1970, Sanger began wanting to sequence DNA as well as RNA. He created a new DNA sequencing method using acrylamide gel-based read-off methods for sequencing. Sanger chose to sequence the single-stranded DNA bacteriophage X174, a bacterial virus. In 1977, Sanger's group completed the first genome to be sequenced, consisting of about 5,375 nucleotides. Once again, he received a Nobel Prize in chemistry for DNA work, but this time he shared it with Paul Berg and Walter Gilbert. He then became the fourth two-time recipient of the Nobel Prize ever. In addition, Sanger also sequenced human and bovine mitochondrial DNA. His contributions established the basic genetic principles utilized by almost every biotechnology application. Sanger's discoveries also allowed us to determine the structure of any protein and analyze its function to map the human genome.